Okay, hi there, welcome again. It's Mr. Phillips again recording the coronavirus science lessons. As the schools are closed, I want to try and allow my students to, prepare themselves for year 13 in this case. Or if you're from another school and you're doing the same, fair enough. But hopefully you'll find this video useful. So what I'm going to be looking at today is continuing year 13. If you're doing AQA, it's topic 5. If you're doing OCR, I've forgotten what the topic number is, but it's at the start of year 13 content. So some questions there that you should really be able to answer based on my last video. Sorry, just a second. But effectively if you want to use those video questions, please pause and have a go at them. Otherwise, we're going to move on and we're going to look at today the first stage of aerobic respiration. So the first stage of aerobic respiration is called glycolysis and it involves coenzymes, which we have to know about. If you're not quite sure by the end of today's video how coenzymes work, it will come much more obvious when I do the Krebs cycle and oxidative phosphorylation, which will be the next two videos I record. So just to give you an overview really, because we're only just starting this, there are 3 stages of respiration that we need to know about. There is glycolysis, okay, which is basically glyco. So think about the word sugars are all like glycogen and things like that. And lysis, it means sugar splitting. Okay, and this forms 2 what we call intermediate compounds, which is a reason why the respiration equation you learned at GCSE is too simple. It turns into something called pyruvate. Okay, and this is actually the part of respiration that doesn't happen in the mitochondria, which is why bacteria are able, even though they are prokaryotic cells and don't have a mitochondria, are able to carry out respiration, because this is the only stage of anaerobic respiration, and it is obviously the first stage of aerobic respiration. So if you draw a sort of line there, both of the stages below only happen in aerobic respiration. Glycolysis happens in both anaerobic without oxygen and aerobic respiration in all living things. Then we've got the Krebs cycle, which we'll do next time, which happens in the matrix of the mitochondria, and we won't mention too much about that for the minute. And there's something called the link reaction that links both of those up. And then finally, we've got something called oxidative phosphorylation, which happens in the inner mitochondrial membrane, and that's where most of your ATP is actually made, and it happens across something called the electron transport chain. But if you're asked what the reaction is called, it is called oxidative phosphorylation. And I think it's worth noting that when I go through the glycolysis reaction in a few minutes, and when we go through Krebs cycle, a lot of the time we'll be talking about hydrogen ions, okay, so H plus ions that are released from the glucose, okay, and in this case it's hydrogen ions that are released from the pyruvic acid, which we'll talk about next time. So the reason when I'm mentioning this now is because at the end of this video you won't know what happens to them. It won't be till the end of the oxidative phosphorylation video that you do, but what you need to understand is that these hydrogen ions are being carried by NAD, which is a coenzyme, and they are being taken to this final stage here to make all of the ATPs that are going to be made, or most of the ATPs is going to be made. And the same from hydrogen from this stage is also being shipped to that stage there. So when I'm saying H plus is being made, that basically is going to be something you won't find out about till later on. So then, this is basically what we're going to be looking at. So I'm going to be talking about this in much more detail, but this here is what's called glycolysis. So what I recommend people do who are listening to this for the first time and not using it for revision, or even if you are using it for revision, is pressing pause on the video now and having a go at these 2 tasks. Because what you need to be able to do is basically explain what glycolysis is. So you need to be able to maybe look at this video and actually try and explain what is happening. So just turn this into sentences. So if you think about it like sentence 1, sentence 2, sentence 3, and then when I go over in a few minutes, you can check whether your understanding of it from the diagram, whether you can interpret the diagram, has helped. It'll only help you initially roughly, but it might just get you really thinking about it, so understanding. And then we need to also know what the products of these reactions are. So anything coming off this flow diagram are the products. So before we start then, we've got the mitochondria, which is where all of this energy is being released as ATP and made as ATP. But actually this reaction isn't happening in the mitochondria. So the Krebs cycle reaction is happening here effectively in the cytoplasm, and then the products of the Krebs cycle will be shipped into the matrix down below. So two terms that we need to know just before we start off. From the GCSE, you've probably done oil rig at GCSE, which is how I learned it in chemistry, and that is oxidation is loss of electrons, and in this case we'll be talking about hydrogen ions, so oxidation is loss of hydrogen ions, and reduction is gain. And basically one always has to occur the others. When I'm talking about something being reduced, I'm talking about something that is gaining hydrogen ions, something else must have given it those hydrogen ions, so that thing is being oxidized. So oxidation is loss of hydrogen ions in this case, reduction is gain in hydrogen ions. And we're going to talk about two coenzymes today. First one is NAD, which is here. You could be asked to learn the structure of it, so it wouldn't be something I'd recommend you learning just yet, but as you get close to year 13 exams, you need to know the structure of that. So it's got a nicotinamide molecule, two ribose molecules, sorry. It's got phosphate times 2, and it's got an adenine. And its job is the most important thing, is it helps, it is reduced, sorry, so it helps oxidize molecules and making them smaller and taking up to 2 hydrogens away for later on. And we'll talk about that and it'll become a bit more clear later on. There's another molecule which we've got here called coenzyme A. We won't be talking about it today, but it's another we're called coenzyme, so we'll be talking about those later on. So then, let's look at glycolysis. So glycolysis, what do we need to know? It happens in the cytoplasm. We've said that. It is the first stage of aerobic respiration, which you should know means with oxygen. It is the only stage in anaerobic respiration. And just want to really point out that anaerobic respiration means no oxygen. Some people fall into this gap of saying it forms less oxygen, but it needs no oxygen to anaerobic respiration. And you get a lot less energy or less ATP when you do anaerobic compared to aerobic respiration. It's an ancient biological pathway, so that means all cells, prokaryotic cells, eukaryotic cells, all do it to release some energy from the glucose molecule in the form of ATP, as you're about to find very little ATP. You only really need to know the outline of the pathway, but I would always suggest being able to draw a flow diagram of it and talking it through and writing it through in detail. So this is probably a slightly more complicated version you need, but I like this one because it means that if you understand this, you'll understand it all. So we start off, what you start off with then in the cytoplasm is your glucose molecule. Now glucose is not very reactive. So when we did about ATP last lesson, last slide, I said that sometimes things get phosphorylated, and what that means is it makes something more reactive. So what we actually do, it seems a bit weird because we're trying to make ATP, is we actually use one molecule of ATP that is converted from ATP adenosine triphosphate into adenosine diphosphate, and that phosphate molecule that's come off has been put into, so imagine that in there, into the glucose to make glucose 6-phosphate. And what that means is that that phosphate is attached to carbon number 6 on that glucose, making it more active. Now what then happens is the glucose molecule, which now become more active, gets converted from glucose 6-phosphate into fructose 1-phosphate. And all that means is it's being turned by an isomerase into an isomer of glucose. So fructose has got the exact same formula, there's only 1 phosphate still, it's just now on a different carbon, just on carbon 1. They then use another ATP molecule, okay, converting the ATP into again an ADP, and the phosphate goes in and it becomes a 2-phosphate structure, which you can either call fructose 1,6-biphosphate or hexose 1,6-biphosphate. Doesn't really matter, there's a couple of different names for it, so whatever name you've got in your exercise book and your textbook is fine. Till this point, this stage is called the phosphorylation stage above the line. And what we've actually done is we've actually used 2 ATP. So think about it, we've already got a debt effectively of 2 ATP molecules, and that's what we're actually trying to make in respiration, because this is, don't forget, our universal energy carrier. So how do we use it? What do we do then? How do we move it forward? Well, let's go forward. So that is a hexose 1,6-biphosphate is a 6-carbon sugar, don't forget, because it is it's come from glucose, which had 6 carbon, and it's gone down to fructose and then to that. So now what happens is the molecules split into 2. So the molecule splits to make 2 loads of triose phosphate, okay, and then triose phosphate is a very unstable structure. So the triose phosphate then is broken down through a series of intermediate products, which you don't need to know what those intermediate products or compounds are, and then they get turned eventually into 2 pyruvate molecules, and this is where oxidation occurs. So the triose phosphate molecule loses some hydrogen ions. So if you remember what we said earlier on, oxidation is the loss of hydrogen. So those hydrogen ions move from the triose phosphate and attach themselves to the coenzyme NAD to make what we call reduced NAD. And that just means basically that the NAD has got hydrogens attached to it. Okay, and those 2 reduced NAD then go off to the final stage of respiration, which we'll talk about in a few videos time. Then what else happens as it goes up from triose phosphate down to the pyruvate is there are phosphate molecules released from these triose phosphate and released from the intermediate compounds to form ATP, and this is called substrate level phosphorylation, because what is happening is you are making ATP as you go from one substrate to another substrate to another substrate. So substrate level phosphorylation is occurring. And if you look at this, we've got 2 ATP being made at the top here, or middle here, and 2 ATP at the bottom. So that gives us I suppose a credit, if you think of it like that, of 4 ATP. Okay, but obviously if you look earlier on in the reaction, we've used up 2 ATP. So if we look at what the products are, and this sometimes is overlooked by students, or they make simple errors here that they shouldn't make. Okay, what we've got is a couple of different products. We have got pyruvate. We've got 2 molecules of pyruvate as a product, which will then used in the next reaction on the next video. We've then got 2 molecules of reduced NAD, which means NADs with hydrogens on it, and the hydrogens have come from oxidizing triose phosphate. Okay, but also we've got products of 2 and then 4 ATP. So depending on what question you're asked will depend on what you need to say. So if we were to look at the actual products, this is what we have. Depending on what the question asks you, you need to be quite careful. So there is a yield of 2 ATP. So whilst you're actually making 4 ATP, because of that there and the fact you've used 2 ATP up, you only get a yield of 2 ATP. So not a lot of ATP altogether there. And then you get a product of 2 reduced NADs, which means they've got hydrogens attached to them, hydrogen ions, and 2 molecules of pyruvate. Okay, and then obviously what we're going to look at is, well, what happens to these in the next video. So the ATP might be used to help move the products themselves into the mitochondria. Don't forget, this is all happening in the cytoplasm. NAD has to go into carrying the hydrogens into the mitochondria for the final stage, and the pyruvate molecules have to go into the mitochondria to go to the next stage called the link reaction and the Krebs cycle. So there you go. So again, if you wanted to see what you could do, you could pause this video and do some of these tasks, and that might help you. And again, for my students, you'll be able to access this original PowerPoint as you need it. There's some questions you can answer there, and again, I'm not going to go over them because of time. I want to get the video as quick as I can. But let's have a look at some of these. So correctly answer which of the following molecules is glucose. So if you look at those, pause it if you're not sure. The correct answer is C. It'll be beta above. So if you remember from the year 12 stuff, OH is above it, so it'd be a beta molecule above carbon 1. Then we've got which is the first part of respiration, so you should know that is glycolysis. Where does glycolysis take place within the cell? You should know it is in the cytoplasm of the cell. What is the energy needed for? So energy is needed for both B and C, anabolic reactions and the movement of cells. What happens to glucose during the first pass of glycolysis? So glucose needs to be phosphorylated because it needs to become more active, reactive. And then how many phosphates make up ADP, adenosine diphosphate? So that is 2.